we had a chat quite a lot of years ago now, and I was interested in white eyes, and white eyes are a really speciose bird group, and we got together and we had a PhD student doing work on the relationships of African white eyes, and then after that PhD, Julia had another master's student who continued this work, and we've gradually been putting together an overview of African white eye relationships, evolution, and so forth. And we'll hear a bit more about this from Julia now. So over to you, Julia.
Okay, well, thanks, Robert, and thanks for inviting me here tonight. As Robert said, I'm going to be sort of talking about this group of birds, white eyes, or technically what they refer to as Zosterops. So, we have 10,000 species of birds on our planet, roughly, which are represented here in this phylogeny. We can see the sort of evolutionary relationships of all the birds on our planet, which has been generated using molecular data. Why is it that evolutionary biologists seem to prefer some lineages of birds to others? So, for example, the paleognathae, which include things like emus, ostriches, rheas, cassowaries, are perhaps not so interesting to evolutionary biologists, although maybe to some, because obviously they're here at the base of a radiation of birds. But there's other groups of birds. We can see, I haven't got a pointer, but I am going to jump out, sorry. Things like the ovenbirds from South America, or the finches over here, and the white eyes, the Zosterops, which occur here, which are incredibly fascinating for evolutionary biologists. Now, why might that be? This figure gives us some indication, because it's not just showing the evolutionary relationships, it's also showing us the diversification rates of these birds. So the diversification rate is a speciation plus extinction. And what we can see with some of these birds, like say the ovenbirds, that the rates here are very, very fast. So these are the groups that are in sort of coloured in the orange and reds and the yellows, and the birds like the paleognathae, which are sort of much slower regarding their speciation, are colour-coded in the dark blue. Obviously, for evolutionary biologists, it makes sense for us to look at these very young, rapid radiations, because it sort of helps us look specifically at speciation, and it avoids these complicating causal events that we might get from looking at these older radiations. Okay, so before we start talking about Zosterops or Zosteropidae, let's have a look at a few groups that evolutionary biologists are really interested in. So I suspect you're all really familiar with Darwin's finches, and anybody who studied biology will of course seen this as this classic textbook example of a radiation. And we can see clearly with Darwin's finches, and here we have the lovely large sort of brown finch. If we look again at this phylogeny over here, we can see clearly that the author has put these pictures of the birds' heads and beaks on the phylogeny. You can see that there's huge sort of disparities in the shape of the beak. Okay, so you have three lineages in Darwin's finches. You have these brown finches here with different shaped beaks. We've got, and they all feed on the ground, and they feed on seeds or seeds and cactuses. You've got another lineage, I don't know why I've come back over here, this one here. These are the tree finches, and as the name suggests, they inhabit trees, and they also, these guys feed on insects. There's also a vegetarian finch, and we see at the base of that phylogeny, there's a warbler finch as well, and the coccus finch. Okay, so what's going on with this radiation? Well, clearly ecology is really important, and they diverge in their niches according to not only the environment they live in, so you've got the brown finches and the tree finches, but also their diets. So you can see quite clearly from those lovely diagram up there the different shaped beaks. So in biology or evolutionary biology, this is termed an adaptive radiation. And if you're not familiar with the term adaptive radiation, this is an evolutionary process that can cause a single ancestral species to rapidly diversify into a multitude of species that then exploit various ecological niches. And they differ, don't they, in their traits. That might be a morphological trait like we've seen, the beak of the finch, or a physiological trait. So adaptive radiation is like really kind of important for evolutionary biologists because it's thought to be responsible for much of the tree of life's eco-morphological diversity. But you can get non-adaptive radiations, okay? So this is when you have like a proliferation of species accompanied by negligible or infrequent ecological differentiation. I'm just going to skip over that. Here's another lovely example of adaptive radiation. These, again, are finches, so there is something about finches that they radiate. We can see with the Hawaiian honey creepers, again, if we look at that diagram up there, we can see all these different shaped bills, and they're much more dramatic, actually, than Darwin's finches. There's more species, so we have around 50 species of Hawaiian honey creepers and just 14 species of Darwin's finches. But they've radiated over similar time frames, so sort of 5 to 6 million years. And then finally, just one last example, because I'm talking to bird-orientated people, and this is actually one of my favourite examples of adaptive radiation. These are the Madagascan Bangers, and this is a paper that came out in 2012. You can see, very interesting paper, they have this lovely figure in the paper. You can clearly see these different bill shapes, obviously all relating to different diets, and we can see that with these guys here, they've not only diversified into different habitats on Madagascar, so in Madagascar you've got these sort of tropical rainforests to the east and then sort of drier forests to the west, but they've also diverged in body size. You can sort of see that from that diagram. So here's some examples for you for adaptive radiation. Okay, so we've looked at these birds with very different morphologies, but what's so interesting about white eyes? Well, I'm going to tell you. So we've already kind of had a little sort of precursor to that when we look at these rates of diversification. So very, very rapid rates of diversification. And some previous authors, so in the paper by Moyle et al., which actually included Gerard Diamond, who some of you might have read his popular science books, they found that with invertebrates they had one of the highest diversification rates, not the highest, that belongs to another group of vertebrates I work on, cichlid fishes, but an exceptionally high rate of diversification. Okay, so just before we talk a little bit more about Zosterops per se, let's just have a little look at Zostropodidae, the systematics of the group. Now, it's a family that contains 140 species, which is super impressive. But you'll see from this list here, and I've just taken this from the Handbook of Birds of the World Alive, that most of that diversity is within the genus Zosterops. So all these other genera are quite species poor. We can see that quite a few of them actually are just monotypic, so just a single species in those genera. Interestingly, I note that the hyenas and the babblers, which we can see illustrated from these beautiful paintings from the birds of the world, are now placed within the Zostropodidae, okay? But actually, when we look at the rest of the group, they're quite homogeneous, aren't they? So all look quite similar. This one's a little bit of variation. I've just included an example from each of these genera here. Okay, and we can have a quick look at some of these guys. So let's hope that these videos work, which I downloaded shamelessly from Birds of the World. This is the giant white-eye. Okay, amazing bird. It's quite a bit bigger than the normal white-eye, so typical white-eye. So it grows about 14 centimetres. And this little guy is found sort of endemic to some Pacific islands. So when we compare that bird to, let's just get the typical white-eye. Hopefully this will work. Yep, okay, it's ready. So this is like a typical white-eye. They're quite pretty little birds, so I guess some of you probably have come across them on your travels. You can see they're sort of quite uniform. They've got a sort of greenish back, a bit of yellow. Some of them have got pale bellies, some of them have yellow bellies. And of course, what gives them the name is this lovely white eye ring that in some species is quite distinctive, like this guy here. This is the oriental white-eye Zosterops palpabrosus. Now, compared to those examples I gave you of adaptive radiation, which obviously you will have noted were from islands, so very spatially small systems, Zostropody have this enormous distribution, and you can see that they occur across Africa. They're also found in the associated islands, so the Gulf of Guinea and the Indian Ocean island, Madagascar, through to Arabia, India, and then they're found all throughout Asia and down to New Zealand. Someone showed me one of the silver eyes that they've seen in New Zealand. And it's kind of interesting because 90% of Zostropody diversity is actually found, or they're actually found on islands. And a lot of that diversity is found in that sort of region in Asia. So these guys have an exceptional colonisation ability. And we can see, again, this is going back to the Moyle et al. Paper in PNAS, but it's actually Zosterops, it's the genus Zosterops, which has this huge range. So this is their phylogeny, and this includes probably about, 30% of all the Zostropody. And you can see that Zosterops themselves have this huge range, whereas those other genera have quite contracted ranges, or quite small ranges comparisons. And you can see that they've also included the euhenas there as well, which interestingly don't form a natural grouping. So again, unlike these previous examples of adaptive radiation, white eyes are really homogeneous, okay? They all look very, very similar. And these are some gorgeous plates from the Handbook of the Birds of the World. And we can see, yes, they look pretty similar. They're incredibly hard to identify in the field, and taxonomists have had an awful job trying to resolve their relationships. So we can see here that, you know, a bit of yellow on some, they've all got these sort of olive backs, some have a grey belly, some have a yellow belly, some have eye rings, some don't have eye rings. You can go through, oh yeah, here's some nice sort of white belly forms, some yellow belly forms. So these are all Zosterops found across their range. Again, here's some more. Some of the African ones with this sort of larger white eye ring. And then finally, we do have a few slightly different looking birds here. Now these guys were formerly placed in a genus called Spiriops. And some of these guys, they're African. They're found in Gulf of Guinea, for example. And they do look a little bit divergent. So that's it, okay? So that's what we're having to deal with, a really homogeneous, species-rich group of birds. And the other interesting point with these birds is that, unlike those examples of adaptive radiation, where birds are huge specialists, so you'll have birds that are just feeding on one sort of dietary, in one dietary niche, these guys are, we think, are ecological generalists. And the reported diets, when I looked at this Handbook of Birds of the World, suggests that they feed a sort of mixture of insects, fruits and nectar. So yeah, so essentially you can hopefully see, just from looking at these plates, that previous taxonomists, people like Meyer, Mies and Reginald Moreau, who actually did a lot of work on the African white eyes, were really struggling with their taxonomy. And I kind of love this quote by Ernst Mayr. So he says, "I know of no other group of birds in which close relatives," and he gives some examples, "may differ more from each other than they do distantly related species. Indeed, some oriental species are almost indistinguishable from African forms, from which they must have been isolated since remote times." So he's also then, back in the 60s, before molecular genetics, are really struggling with the taxonomy of this group. Okay, so we've got this radiation, very, very species rich, but incredibly similar. So we can sort of really ask, like, why, what's happened, what's driving the diversity of this group of birds? And I'm not really sure I'm going to be answering that question today, but hopefully I can tell you some of the findings and something about the evolutionary processes of this group. Okay, so we sort of became interested in African Zosterops, a little bit what Robert said. I was working in Africa and some other groups, and I'm a big birder, so I've sort of seen these guys and kind of thought they could be quite interesting. And this is some previous work that Robert's student Ben Warren conducted. So he was looking at the Indian Ocean Island birds, okay, and I'll come on to like his most important findings, but for me what was quite interesting was, I'm just sort of highlighting two areas in that evolutionary tree. He not only showed that Africa wasn't forming a natural grouping, so what do I mean by natural grouping? That doesn't include the ancestor and all the descendants, but he also included some subspecies from Africa. So you can see the there's some trinomial names in that phylogeny. And for example, Polyagaster, was a described species from Africa. And you can see that Polyagaster is in both of those groupings. So there's something strange going on here, and it might allude to the fact that there's more diversity than we initially thought when we look at the molecular genetics of the group. Of course, the most important finding of this work was the fact that the Indian Ocean island birds had probably colonised the Indian Ocean at least several times. I'm not going to go into that today. Okay, so our earlier work, which was conducted by Siobhan Cox, who, as Robert said, was our joint PhD student, we focused on these African montane birds, the Polyagastrus. And these birds are actually quite gorgeous, they're quite big, they're found in the Sky Islands, so they occur, we can see in the red dots here, from Hare Mountains in Tanzania, through the Tata Hills, Chula Hills, Mount Kenya, up into Ethiopia. And these birds are just found in these high montane forests. And they're a bigger bird than the lowland birds, they're heavier birds, and they're kind of brighter in their plumage as well, and they have this really big white eyelids, so they're quite distinctive. Okay? And what we showed in that piece of work with using molecular genetics was that these lovely highland populations are more closely related to the lowland populations that occur in savannas than they are to other highland populations. So we have this repeated transition from highland to lowland, okay? And this would be, well, we interpret this result as suggests that it was explained by ecology, so niche divergence, so they're changing altitude, they're changing habitat. And that's opposed to the other alternative would have been that you have a lowland bird that sort of colonises the highland and then it diversifies within the highland habitat, okay? But one of the main points here was really that because of the genetics, it suggested to us that these highland birds are probably really good candidate species and they should be elevated to species status. So what you see here are all the subspecies that are found within these highland regions. And since we produced this piece of work, I see that the birds of the world have now elevated Polyagaster to these different species that we see here. And in fact, other people have now looked at the call data for these birds and suggest that the call data from these different montane massifs is actually, they actually do have different calls. Okay, so on to sort of the current work. So one of the things after we found this surprising result was that we decided that it would be good for Siobhan to look at all the African subspecies, okay? So unfortunately we couldn't sort of send her out to the field, or we couldn't go to all these places because we can see from this map that they occur obviously pretty much all over Africa. But what we did was sort of raid the Natural History Museum at Tring, courtesy of Robert being there, and we were able to use these amazing collections as archival material to extract DNA out of this material for our phylogeny. We also had people sending us blood samples as well. So with this map here, you'll see there's 4 different colours, 4 different ranges. And we've already talked about what was Polyagaster, these montane forms. We already know that there's probably more diversity there. But in this study, we also were able to include a few other species that we didn't previously include in that initial paper. Now you'll also see this range of the yellow bird, and this is Senegalensis. Now Senegalensis is from the lowland. You can see its range is huge. It's just found pretty much all over tropical Africa, apart from obviously the Congo Basin and southern Africa. and there's 14 subspecies of Senegalensis. And I can tell you they all look pretty much the same. I've looked at them in the Natural History Museum, and it's incredibly hard to see sort of differences. But there are, the previous taxonomists did find some subtle plumage differences, which is interesting. And then finally we have, or I shouldn't say finally, penultimately we have Abyssinicus. So Abyssinicus is this grey-bellied bird, and it's also got quite a restricted range, hasn't it? So it's found sort of in the Horn of Africa and also in Arabia as well. And finally, just to point out, there's 2 species that have been described from southern Africa. You've got Capiensis, which is the dark blue there, but it's found sort of in the Cape region and beyond, and then also Pallades as well. Okay, now I'm not expecting you to read the phylogeny that I'm going to flash up next. Oh, isn't that awful? So it's really difficult trying to depict phylogenies for people to examine. Right, but you don't need to worry about the writing of the birds, just need to worry about these colours. So hopefully you can see the colours quite nicely from that. Okay, so with this phylogeny, we included everything, so all subspecies from the African continent and its associated islands, so the Gulf of Guinea, the Indian Ocean islands, and like Ben Warren had worked on, Martin Nilo. We also included a whole bunch of outgroups. And what are outgroups? Well, that's all birds, not all of them, but some birds from Asia, because obviously if we want to focus on our African group, we want to perhaps look at colonisation, we need birds outside of that range. So what I've highlighted there in that rectangle is our African radiation, okay, so, or I should really call it like the Indo-African radiation. And what we showed with this phylogeny, it's built on molecular data, is that all the mainland birds and these island birds, so from the Gulf of Guinea here and from the Indian Ocean island, they're all sort of forming one natural grouping, okay? So that was one result. But the result that perhaps is interesting is that these original species descriptions, these birds like Senegalensis and Abyssinicus, they're just found all over the phylogeny, aren't they? Just peppered all over. So you can see the yellow squares are just falling out everywhere. And this suggests immediately that they're not natural groupings. So whoever described Senegalensis got it sort of vastly wrong. And, I mean, it's understandable because these birds look so similar. We can also see that Abyssinicus, we have a sort of grouping here, we've got some Abyssinicus there, and more Abyssinicus there. So there's some things going on with this phylogeny, it's quite interesting. Okay, so we've already talked about Polyogaster. We know Polyogaster is not a good species, and actually the subspecies is a good species. And generally this new work that is currently in review, we find that highland and lowland birds are each other's closest relatives, but some of the highland birds we didn't include before, which are actually from this region, so from Ethiopia, the Ethiopian highlands, have actually sort of diversified, or they've speciated in the highland setting, so that's a little bit different. Okay, here's Senegalensis. This I think is the most difficult one to work on. So Senegalensis, clearly, as I say, there's lots going on there, and it would seem that quite a lot of the subspecies should be possibly elevated to real species. So that's something I'll come on to. This is Abyssinicus. Abyssinicus has already got one of these groupings has already been elevated to species status. I notice in birds of the world, this yellow-bellied form. But interestingly, I just wanted to highlight a case of cryptid diversity. And what I've highlighted there are these two green circles. And these are both Abyssinicus soctranus, okay? So it's described as the same subspecies. But what are they doing? They're kind of at either ends of the phylogeny, aren't they? So there's a massive, deep genetic divergence between birds that were collected, just go on to the next page, from the island of Sotra, which is here, and also these birds are found on the mainland in Somalia, here. And when we look at the skins from the Natural History Museum, hopefully, I mean, there's no difference, is there? I mean, somebody might sort of say, oh, that top one's got a slightly wider belly, but the differences are, vague, to say the least. And we took some trait data, or Federico took some trait data, and you can see that the triangles indicate the bird from the island, and squares indicate the bird from the mainland. And you can see from this morphometric data there's just no difference, okay? So this is a really surprising case of a sort of cryptic speciation. These birds look exactly the same in the field. I mean, I don't think you just wouldn't be able to identify them. But obviously, genetically, they're very, very different. I suspect if one could get into Somalia, which you might not want to at the minute, then probably the calls are different from these birds. And, perhaps when that country opens up a bit more, that would be something, it would be very interesting to get that call data. Okay, not really much to say about these southern African Zosterops, capiensis and viridans, which are here in the phylogeny, but just to say that sort of previous work has assumed that they were very, very closely related. And we can see, because there's some Senegalensis and Polygaster mixed in to that grouping there, that they're not. And it suggests that, again, with these birds, their amazing colonisation abilities, that they probably colonised southern Africa a couple of times. Okay, so I'm not really going to bamboozle you too much with some of the methods we use, but just to say that we ran our phylogeny, our data, using sort of quantitative methods of species delimitation. So rather than just eyeballing the phylogeny, we are trying to do something in a more quantitative fashion. And essentially this examines the transitions, or these methods examine the transitions through using the phylogeny, and here this phylogeny's got a time element to it, so a time tree. So it uses the tree branching patterns between the long interspecific branches, that's the sort of speciation events, and also the short intra-specific branches, that's sort of population processes. Okay, and this is just like a heat map here, so it's kind of showing the bright clusters are just where the program's sort of indicating that those clusters could be good candidate species. I think it's an interesting method, but of course you do also have to consider the biology of the organisms as well. So what did we find? Well, actually according to the Handbook of Birds of the World, there's previously now 14 mainland species, because it's taken our Polygaster already and elevated them to species status. So there were previously 14, according to the current handbook. We've now sort of suggested there's probably around 25 mainland species. I mean, I'm suggesting that, or we're suggesting that. Obviously, I think with some of these subspecies of Senegalensis that we're suggesting should be elevated, it would be really nice to get a lot more data. I mean, we've only included a few samples from some of these Western Senegalensis, and really, somebody needs to go out and do a hell of a lot more collecting, so you've got sort of much more population level data. Interestingly, I mean, obviously with our phylogeny, remember we're including these island representatives from the Gulf of Guinea and the Indian Ocean, and that's really nice because they're all nicely described apart from a couple of subspecies, and we just sort of indicate there's obviously 18 species currently described, and we're suggesting there probably should be around 21 island species, and that's because there's a couple of subspecies of what's termed Madagascan, and those look like based on our genetics, that they're quite distinct. Okay, so feel like we've done it. We've resolved the taxonomy of African Zosterops just like that. As I said, there's probably still quite a bit more work to be done just to really confirm our findings. Okay, so just quickly moving on to sort of look a little bit more at evolutionary processes. We can take these wonderful densely sampled molecular phylogenies that we generate, okay? We can sort of put a time element on them, so we can produce a time tree, and then we can start to look at things like the diversification rates that you encountered on the second slide of this talk. So just remember that's the speciation and extinction rates. Okay, so it's a little bit technical now, but hopefully you'll follow this. So we can use these things called lineage through time plots, and these methods really use the information on the time elapsed between successive branching events in the phylogeny to say something about the accumulation of species. So going back to our bangers, this lovely example, classic example of adaptive radiation, we get this pattern. So this pattern is called like an early burst model. You can hopefully see you've got this sort of really sort of rapid increase in the rate of diversification, then it tails off, doesn't it? And this tailing off is suggested to be a sort of saturation of the niche space. So the birds sort of, they colonise an island where there's, nothing else to compete with. They rapidly diversify, and then as more species appear, niche space becomes saturated, and that sort of tails off. Okay, but this is also what Moyle et al. Found when they looked at Zosterops. This is the clade B, which is the Zosterops group. And that's kind of interesting because they don't look like they're adaptive to me. They're this homogeneous group of birds. They don't have any differences particularly. And it's kind of interesting because some work came out recently by actually one of my colleagues at work, and they suggest, well, perhaps this pattern might arise in other scenarios, not just adaptive radiation. And maybe one explanation is simply geographic speciation, which kind of would make sense with our Zosterops. And also, we find the same pattern in these North American wood warblers, which, unlike Zosterops, occur in sort of temperate landscapes. So we also found a similar pattern. This is just using some different methods there, so we don't really need to worry about that. But we also found the same pattern as Moyle, looking specifically at our regional phylogeny. And I think that the point I wanted to make here is that you might expect island birds to diversify more quickly than mainland birds because they've got all this available sort of niche space and not sort of in competition like you would be if you live on a continent. But actually, the rates of diversification are very, very similar across the island and mainland birds. But basically, what's more interesting than looking just at lineages is to look at the morphological evolution of the group. So I'm just going to spend the last part of the talk just sort of talking about the island and continental birds. And again, we can look at these amazing collections at the museum, all these birds that they have, particularly have a fantastic collection of Zosterops. And what my student Federico did was he took a whole bunch of trait data. I don't really need to go into that, but 9 different traits of the bird, and he spent quite a lot of time up in tree measuring birds through a pair of calipers. So with all that trait data, we can sort of combine it with the phylogeny, which I'm going to talk about in a minute. But I just wanted to show you these morphospace plots. So just to quickly introduce you to principal component analyses, it's sort of a dimensional reduction tool. So you can take all that trait data and it can sort of reduce sort of a large set of data, a large set of variables to a small set that contains most of the information of the large set. So hopefully that makes sense. But with this plot, hopefully you can see that our mainland birds are occupying a really tight, constrained area of morphospace, aren't they? They're just kind of all plotting together. But when we look at our Afro-Indian island birds, so within that radiation that we were focusing on, hopefully you can see the grey plot there with the triangles, we can see that they've expanded into morphospace somewhat. And this is probably because of these more aberrant birds that I was talking about earlier on. So birds that used to be placed in Spiriops are now nesting deeply within the Zosterops. They're probably causing that expansion of morphospace. And when we look at the Zosteropidae as a whole, so everything outside of that radiation I've been talking about, we can see again there's a much bigger expansion of morphospace. So there's much more evolutionary novelty going on. And of course, if you live on an island, it allows you to sort of basically evolve on new evolutionary trajectories than living on the mainland. Okay, this is kind of technical, but just hopefully you can just stay with me for a few slides and then we're going to finish up. So we can take that very interesting trait data, those PCAs, and we can combine it with our phylogeny, and we can produce a sort of mouthful of these disparity through time plots. So this is something that Luke Harmon developed, and he was working on different lizard radiations. And really all you need to just take on board here is that these plots sort of compare your morphological disparity that's simulated under a model of Brownian motion, which is a random walk, with the observed phenotypic disparity between and within the different clades of your phylogeny. Okay? And that's relative to the total morphological disparity at all the time steps in the phylogeny. These are a bit hard to get your head around, and to be honest with you, I took a while to work out what was going on with these plots. But what I really wanted to show you here, I think it's really the take-home message, is that when you have a sort of negative plot, like we have here for these Australian gametes, this is suggestive of the adaptive radiation that we came across earlier on. So here you're sort of expanding into morphospace, you're filling new niches. And the top plot, the positive plot, obviously the solid line is above the dotted line, is an example of convergent evolution. Now, if you're not familiar with convergent evolution, it's an independent evolution of similar features in species of different lineages. So, for example, this is a classic textbook example of convergent evolution. Obviously, you've got these unrelated lineages, and they've all evolved wings. Okay, so what did we find? The African radiation, we can see, we've plotted the different PC axes there, and if some of you are wondering what the grey blob is, that's like a 95% confidence interval. Hopefully you can see some positive peaks there in that plot. So it suggests that our birds have, essentially we have a signal of convergent evolution going on in our birds, which is kind of interesting. But unsurprising, isn't it? Because I wouldn't have expected to get an adaptive signal from birds that essentially look the same. So I think, we wouldn't have expected to see a plot that was sort of in the negative half of the figure there. Interestingly though, when we compare the islands with the mainland, and we can do this because, we had a very similar number of birds that are just, described from the islands, and now we have a similar number in the mainland. We see that the signal of convergence is really coming from those mainland birds, not the island birds. The island birds are just sort of no different from this random walk pattern. So the morphological convergence that we identified really appears to be driven by these mainland taxa, and it's most likely the result of niche divergence between the highland and these lowland species. And you can see in morphospace there is some divergence, isn't there, when we look at that trait data between the highland taxa and the lowland birds. The trait evolution in the mainland taxa really is identified as one of constraint. I'm not going to really show you that data. So although ecological or ecologically driven phenotypic divergence has occurred between these highland and lowland habitats, species within these habitats only display really quite small variables. And this really probably is ultimately due to fewer ecological opportunities, due to the niches in continents being more greatly filled. Okay, so in contrast, the island taxa trait data is obviously not significantly different from Brownian motion, but they occupy this much larger area of morphospace that we've seen from this plot. And this really indicates the sort of potential release of species competition that has allowed these island birds to continue to evolve novel phenotypes when compared with their mainland relatives. And this pattern's actually been noted in these carduline finches. This is some work by Losos and Ricolets. And they just show that the continental birds are sort of quite constrained from this plot here, but actually the Hawaiian honey creepers, so that's obviously like a subset of these finches, as we saw before, have this huge area of morphospace. So, you know, birds on continents probably have a bit slightly more constrained morphology. Okay, so I'm just going to sum up really for today's talk and just highlight these main findings. So of course we've shown, or hopefully we have shown, there's an underestimation of these cryptic mainland species. We've shown that many of the traditional species groups are not natural groupings, and that species diversity is really underestimated by up to 50%, which I think is quite impressive. And I've also shown you some examples, particularly an extraordinary case of the Sotra white-eye. So, you know, it's a really classic case of cryptic speciation, where you have birds that look exactly the same, but genetically they're completely different. They're not each other's closest relative. We've also looked a little bit at some evolutionary processes, and we've seen that the mainland birds, it was suggested that there's convergent evolution, and this is possibly driven by these ecological differences between the highland and lowland birds. And though I haven't shown you this today, because this is some additional sort of evolutionary models, it suggests that sort of constrained as opposed to adaptive processes have occurred during the radiation of those mainland birds. And then finally, just to finish off, we've seen that in these island birds, unsurprisingly, there's probably been ecological release, and of course there's greater novelty in some of those island birds. I just want to finish off by thanking the gang of people who helped this work, particularly Federico Martins. Unfortunately, he couldn't be here tonight, so he's out doing some badger surveys. But Federico was fantastic because, you know, he came to the lab, he wanted to work on bird genetics. I just gave him the hardest project that we had in the lab. So you know, he's been really great to sort of deal with all that genetic data. And of course, Siobhan Cox, our previous PhD student, she's out of academia now, but you know, she obviously produced a lot of very interesting genetic data for us. So I'd just like to finish off by thanking you all for listening to me, and I'll take any questions.